Hello everyone, this is Wenbo Wu from University of Delaware, a current graduate student in Joe Rosenthal's group. I'm very glad to introduce X-ray photoelectron spectroscopy, or briefly XPS, in our department to you. Let's start from some simple physics. In the XPS, we use an X-ray beam to irradiate samples and kick out the inner electrons, followed by the measurement of the kinetic energy of those electrons. Since the energy of the X-ray beam with a particular wavelength is known, and the energies of the ejected electrons are measured. By using the equation on the right side, we will be able to determine the binding energy of those electrons. Upon knowing the binding energy, we will be able to get the infos about the element, orbital, and chemical state of the atom from which the electrons are ejected. This is a photo of our instrument. As you can see, most parts are covered under metal pieces. People can simply sit in front of the computer to set up the parameters and collect the data without worrying about the X-ray source. This is the front view of our instrument. If we take off all those metal coverage, it will look like this inside. The electrons from the source are focused onto a metal anode to generate the X-ray beam, and then irradiate the samples. The ejected electrons travel through an atomizer and finally reach the detectors to present the XPS spectra. This is an example of the XPS spectra. This is in a recent published work from Joe Rosenthal's group. We took XPS spectra of the rose metal before and after CO2 reduction reactions. In this way, we will know the detailed metallic components of our electrode surface. This is pretty much everything I would like to share with you today, and I'm looking forward to seeing you in the future. Thank you.
Hi, my name is Carly Byron. I'm with the TOF group at the University of Delaware, and I'm going to talk briefly about the TOF-SIMS instrument that we have in the surface analysis facility. So just to give a very brief overview, TOF-SIMS stands for Time of Flight Secondary Ion Mass Spectrometer. It's a surface-sensitive technique where you bombard the surface with an ion beam. And then those beams, those ions hit the species on the surface, and they eject secondary ions from the surface up through these transport optics. And the mass of the ions that were ejected can be identified based on their time of flight through this tall vertical chamber up here. And those times of flight can be turned into a spectrum of masses. So I have a few pictures here of the TOF-SIMS that we have. It's on this sort of desk setup. Here on the left are the computers that we use, where you can control the parameters of the instrument. There's a joystick where you can move the sample holder around. And then here you can see the sort of vertical chamber where the ions travel up and then down, and you can get the time of flight. This all happens under vacuum here. This is the vacuum chamber. And it has to be under vacuum because so that the ions can travel unimpeded. And then this is like the chamber where you insert the sample. Here's like a sample rod, and you insert it into this chamber first, pump it down, and then next into this chamber. So this is just another picture, kind of a closer up view of all the different things that are connected to the instrument. Most of these are to different like gauges and things like that to maintain the vacuum, which is very important to maintain. And then for just a little look at what the data looks like. This is data that was taken from my coworker, Masa, in the Tepelkov group as well, where you can see, actually, it's a good example of the different isotopes that you can see by TOF-SIMS, which is pretty interesting, where when you have something like platinum, where she was looking at an organometallic platinum species, because platinum has several isotopes that are pretty naturally abundant, you can see each type of platinum on your spectrum. So it can be very specific, down to like the exact isotope that you're looking for. So it's a very interesting instrument. it's pretty cool. And if you come to University of Delaware, this is something that you could use.
Hi, my name's Rachel. I'm an analytical student in the books group here at the University of Delaware. One of the instruments that's available in the surface analysis facility is a Raman microscope coupled to an AFM. This is an instrument that's available for students to be trained on or for groups to send us samples to analyze. Let's see. Oh, there we go. It's comprised of a Hariba lab RAM coupled to an Omegascope atomic force microscope. So each side can be used individually or they can be combined. On the Raman microscope side, you can collect Raman spectra of bulk samples, confocal Raman images, and ultra-fast Raman images when using SWIFT, which is just a special detector setting. To use the sample, to use the instrument, you place your sample here on the motorized stage. There, the sample is irradiated by one of the 3 lasers, and the backscattered light is then collected through the confocal optics and directed towards the detector over here. This results in Raman images with a spot size limited spatial resolution of around 2 microns. The AFM side of the instrument can be configured for temperature-controlled liquid measurements, air or conductive AFM experiments. But the most exciting use of the instrument comes when you combine the 2 together for tip-enhanced Raman spectroscopy. When combined, you can collect Raman images with a spatial resolution of around 10 nanometers. So in order to do this, you have to focus the laser onto the AFM tip, which causes a local enhancement of the electric field, resulting in a near-field Raman scattering. This is really cool because once you subtract out the far-field scattering, what you're left with is a Raman image that has a spatial resolution that's well beyond the diffraction limit. And then you can couple this to the topographical information that you're already collecting using AFM. So this is an emerging technique that has been gaining a lot of traction lately, with applications ranging from 2D materials up to biological samples. Thanks.